Video come back in. This is what you're looking at, watch lead? Understood.
That might just be one that's closed up, and that would tell us something about the species, too.
Yeah. Unicella is what Scott France is suggesting. Something like what we collected last week, so that would be interesting to compare. Through video analysis, we've sadly filled up our bio boxes for collections.
Usually that makes the scientists happy when they got their boxes full.
Oh, it does make us happy until we see something else that we really don't know what it is.
We had 10 bio boxes.
Video is clear.
Thank you, pilot. That's great.
Next video.
Copy that.
Well, it seems like there are more sponges than live Lophelia here.
Yeah, I'm a little bit puzzled because a decade ago or so when I was here, there was more live coral on the top of this, but we might not be in the same place. This is a fairly large feature, and that's something else that even if there are some dives that have been done on one of these features, you only get to see so much of it at any one dive, and so it could look totally different on the north-facing side versus the south or east or west.
Like a 180, huh?
We're on the inland side of the mound, so it might be very different on the outer part as you go into deeper waters. I don't know if the mound is lower.
Sounds good.
Is shallower or deeper as you go farther offshore. I think it's about the same height, though. Seemed like that was on the bathymetry.
What do the coral experts think? Is it possible any of the water chemistry or the oxygen content may also impact
meters of 180?
How much grows or lives in this area?
We got lots of
over
time, so
decades or centuries.
Well, I would imagine that this part of the water column would remain fairly well mixed and wouldn't be oxygen poor. And I think that the ocean chemistry would be okay here for corals.
15
I guess
meters,
there's
that's
a cruise coming up next
meters,
month
bearing
that is going to study that
speed
a little more carefully. Any other ideas from other scientists on shore? Or?
Good copy.
You come in. Yes.
It's like another conger eel.
Yeah, you can call out.
Copy, bridge, thanks.
And you can see that they really like this dead coral matrix.
Does anyone mention why there's an abundance of squid up by Sirius?
I'm not sure. Could ask watch lead about that.
So many.
Watch lead, this is Nav.
Go ahead, Nav.
Yeah, Copilot was wondering if we've had an explanation as to why there's so many squid in this area.
So many squid in this area. Let's see, Mike Vecchione actually, when he called in earlier, said that he's seen much bigger schools of squid. So
hundreds.
I think that they're a fairly common squid, and
You think those?
And I guess they're fairly common in this area.
Okay.
And the wreckfish that we've been seeing supposedly eat squid, right? When the squid come down.
Yeah, the squid feed in the surface waters at nighttime, and then during the day, they don't want to be the subject of another larger organism's dinner. So they head down into the darker waters, but often in these areas, they don't realize that the wreckfish are sitting waiting for them to descend during their diurnal migrations. And most of the wreckfish that we have seen have been just perched in the gullies of these features here.
Oh, here's a shark.
Come in, partial.
They're one of the chain dogfish, maybe?
No, that looks like the...
Or is that the cat shark? Yeah.
Shark. Oop. We keep seeing them run into the coral because they don't know where they're going with us here. They're blind. Well, they're blind to begin with, likely, or
well,
can they
actually, from what I've been reading, they have really sensitive eyes. So there is a little tiny bit of light that's available down here, so they probably can see at least a tiny bit. But obviously not when we blind them. If their eyes are that used to darkness, this is going to be a little bit rough on them. The crab tried to catch a shark.
We just had a question from, I guess, the social media audience that asking if squat lobsters can close their claws. They always seem to be open, and I think we just saw that actually right on cue. Yes, they certainly can. We've actually seen them feeding in the past,
grabbing little fishes, or
that's just their threatening stance, letting anyone who's near know that they are ready to use that claw or those claws.
Pilot, this is watch one.
Go ahead, watch one.
Could we zoom in on one of these furry sponges here? We've had a request. Thanks. I guess furry isn't the right term, but...
Iris Sampaio and Joanna Xavier are cringing to hear us call them furry.
Spicually. Is that a word?
Spickly.
All right, video coming on sponges.
There's a little tiny white octocoral behind the squat lobster.
Squat lobster looks like he is presenting the sponge to us.
Copy.
Go ahead, pilot.
You can come in a little further.
Now you can see the inside.
Is that the view you wanted, watch lead?
Yeah, that's great. Thank you.
Yeah, no problem.
And we've done several zooms on these and haven't seen anyone living on the inside, although there likely is in some. We just haven't caught them. But we did see some brittle stars that were living at the bases of these spicules.
Pilot's clear.
Right on the white surface itself, which we hadn't seen before.
And I think that the going genus and species for those is Vasella portalese.
Video swapping out.
190.
Thanks, video. Yeah, 190, somewhere in there.
You want me to spin around to 190?
Sure. Why don't we do another 15 toward 190?
After that, spinning now.
Joanna asked if the sharks that we're seeing
that
are
bridge.
pregnant.
Would like to do another one?
And I guess there's really not a
we'd like
way
to do
to
another
tell,
15 meters. It's 15 meters, bearing 190, speed decimal 2 knots.
Ken Sulak had pointed out that maybe they had just had a nice meal, so...
Watch lead, this is Nav.
Go ahead, Nav.
Just want to let you know we are essentially at waypoint 5, we're 20 meters away, but we are on top of the feature, which means I think we are where we want to be. We have 50 minutes left in the dive. So I just want to let you know we have reached our waypoint 5.
Great. If we can just keep flying around on the top of this feature for, it was 50 minutes or 15?
50. 50
Great.
minutes. Yeah.
Okay. Yeah. Let's just keep exploring, and great that we've reached waypoint 5.
Yeah, and a quick zoom on.
Yeah, video, you can come in.
This is, I believe, what we've collected. So we'll be able to get a good idea of it, but maybe we have some more people online now who weren't online earlier who can help us.
It's great.
And we're thinking it's a hydrozoan, correct?
Yeah, that's the consensus. It's interesting that one anemone is growing on this hydroid in the lower right.
Teetering in the current.
Doesn't seem like very much to grab onto, but
no. You can see the anemones in all stages of retraction here. There's one right in the lower left that is completely pulled in. Looks like a little knob. Some brittle stars still. All underneath these anemones
Copy that.
are where the brittle stars are
Pilot's
clinging. There's still millions of them in the area.
and brittle stars.
Just another couple meters left in the move, Lars.
Copy that.
You can zoom out video. Thanks.
And Joanna Xavier confirmed that the sponge is a Vizela, which Nolan Barrett had suggested earlier. That's great. They're certainly the dominant sponge at the top here.
Mm-hmm. And yes, we are at the top of this feature. We're at 375 meters, and we just passed our final waypoint, waypoint 5. So we must have traveled over 600 meters or so since we reached the seafloor, I imagine. Although we might have shortcut a little bit around one of the waypoints.
Copy bridge, thank you. Move is complete, pilots.
Copy that.
At any point, we've reach the top of this feature with a bit more time to explore, so we're going to keep moving around and seeing if
the current increase on top at all.
A smidge, yeah.
Seeing if we can find those abundant concentrations of Lophelia, which we were anticipating. This area is known as Lophelia Banks and is part of a deepwater marine protected area located off Cape Fear, North Carolina.
I think typically they run at like 14 meg. Oh, that's up to 32 meg.
Tossing on lasers.
Huh.
And judging by the substrate and the reliance of all these living organisms on the dead coral structural framework of the skeletal material, can imagine what damage any trawling would do to this area.
Maybe just a bad reading.
Yeah.
And there's another one of those
it's
white corals. Meg. I think this might be what we were tentatively calling Unicella.
We're just talking about the insulation resistance on the motors do too.
It's like a little sponge bowl behind the
yeah.
Unicella.
You can see that all of these sponges are very clean. They don't have sediment buildup. If they did have sediment buildup, they wouldn't be able to clean it off in most cases, and it would probably be damaging to them. So they would have to be in an area of high current. And the current also provides food, of course, but the high current keeps the sediments from piling up, from settling out. Only the very coarse materials can fall out of this kind of current, and we'll be able to analyze the sediment sample that we took earlier when we dug up some of the rubble that we wanted to use for age dating analysis. But almost assuredly, this sediment is rich with fossil remains of organisms that live in the surface waters, and when they die, their shells slowly settle, even passing through the current, but they ultimately land on the sea floor. And they're made of calcium carbonate. Most of them are of the mineral calcite, but the pteropods, which are the largest organism remains that we find, the largest fossil material, they're made of aragonite, and they are very, they are less resistant
I wonder how close to shore we are.
dissolution than the rest of the sediment material. And so
do you know, Levi?
In the sediment record, when we find pteropods, they are great indicators of low levels of dissolution and good preservation of sediments.
Yeah,
so we use these microfossils to tell us a lot about what the surface water conditions are. For example, planktonic foraminifera are single-celled organisms that make chambered tests, and they're single-celled, and yet they can be very complex in their arrangement of chambers, so we can identify different species. And most of them live in the surface waters, and most of them prefer certain temperature ranges. We have species that like it hot and some that can deal with it being cold, so from tropical to polar conditions. So when we see their test remains, their shells in the sands, when we core down, we can look at change in temperature of the surface waters at a location through time. And that information has given us a tremendous wealth of information about climate change through thousands to millions of year timescales.
Just thought it looked like a good shot. Stop for a second. Yeah, you can come out partial. See if there's anything small that we haven't seen before. Could probably get a short move in, Dan.
How much do you want?
15.
Okay. Well,
what do you think? Go ahead, watch one.
I don't think we need to rush it. You're at 50 degrees.
You can come out video. Yeah. We can talk about it.
Great, thanks.
Dan, you think a sponge like this would stay in the lockbox?
Probably not. But it would. Oh, in the lid?
Yeah.
Yeah, the one with the lid.
Yeah, probably.
Even if we, if it was a little bit damaged, we could tell a lot by the spicules.
Okay. Yeah. Does this sample here look sufficient? You
it looks lovely.
On it,
Lasers on?
So Joanna, we're thinking about putting one of these vasellas into the rock box with a lid.
Do me a favor.
It won't be the most pristine place for it to be, but hopefully it will come up in good enough shape to get some pictures and, of course, DNA samples and have the rest of the organism for morphology.
HPU is ramped up.
We can put it in the forward rock box that has a lid to it. So
rock box, inboard, so I can open the lid.
He won't float away, but he might be tumbled around so much. There may not be much left. We'll see how sturdy he is. It's worth a shot.
Grinding on something.
Okay. You keep it there then.
It's definitely one of the characteristic organisms of this site, so that's also good.
Zoom out on main Zeus, and then he'll wing in and take a look.
Okay, coming in.
Yeah.
Is that far enough?
Yeah, I think that's good.
Not much chance we're going to get a rock today.
On your monitor.
And we'll still have one rock box.
That's right, just in case.
Yeah.
Excuse me.
You could always throw some rubble into the empty rock box.
We actually got a very large rubble and sediment sample. And I know it would be nice to have more, but it would likely go through the slats.
Oh, bummer.
Who knows? Maybe it'll be attached to some. He's got to hold on to something.
I just stick this one under the armpit, right?
Oh, ow. Ow. Well, we might have one.
All right, I think it's mostly intact.
Yeah, it's beautiful. Both the sponge and the rubble looks like they are large enough so they won't filter through the slats. We'll just see what they look like after the pot gets stirred.
Port wing going overboard.
Watch leads, I just want to confirm this is Spec 06 Bio.
Yep, that's correct. Thank you.
Okay, port wing is stowed.
We have Niskin bottles we haven't used yet today, too.
Oh, yeah. Pilot, if we could fire the Niskin bottles, that would be great.
Would we like all 5 of them here?
Yeah, we might as well. Either that or if you want to do 3 now and 2 later, but I'm afraid I'm going to forget, so we could do them all here.
Okay, well, we could do 1 now and then maybe 1 further upslope. Would you?
Or maybe 3 now and 2 later, since we have 5 altogether.
Okay, all right, we'll fire all in the same spot.
Yeah, they'll basically be replicates, so that's an important part of the sampling design for eDNA is to have replication at one place.
Okay.
Gonna go ahead and give you back here.
Would you like us to like move ahead like a few meters or something, or just literally all three right here where we're at?
I'd say right here is fine.
Okay. All right.
Thanks.
Stand by on that.
Okay.
Dropping some waypoints.
Yeah.
Okay. I'll stay here for you, Dan.
Yeah.
You can probably fire it now if you want to, Lars.
You alright with that, Nev?
Yeah, go for it.
Just don't move. All right.
Yeah, he just needs me in my position so that way he can properly drop the waypoint target.
Resetting and firing number 1.
There it goes.
Resetting.
So this is
Firing
spec
number 2.
spec 07 watt is our code.
And resetting.
Yeah, you can look back to the previous dives.
7 watt.
And number 3. And
I imagine that would be 07, 8, and 9 watt. If we do 3.
All right. Let me know when you got those targets dropped, Dan.
Yeah, they're dropped.
All right. Cool.
looks like we're gonna have a full bus today.
So was I.
So what we're doing right now is, or just did, firing what are called Niskin bottles. And Niskin bottles are open tubes that have discs at each end that are held open during the descent, and they are held open through some very strong rubber tubing. And when we are ready to fire the Niskin bottles, that means we send a trigger that releases the tension on those rubber tubes, and the ends both close and encapsulate a water sample.
They were all
So
at
it's almost
1939.
2 liters of water, and we decant that water when it comes to the surface, and we'll be freezing these samples so that we can filter them once we're back on shore. But they are used for all sorts of chemical analyses, as well as looking at the organisms that are living in the water, the microorganisms and algae and whatever is available. Looking at the DNA of some of the organisms as well.
Right, so that's called environmental DNA, and that's basically any DNA that is in the water column here. And one of the reasons that we want to look at it at these coral mounds is to determine what organisms are available for food. You know, we know that they're filtering the water. A lot of these organisms, both the sponges and the corals, are collecting their food from the water column, from everything that's passing by. And so getting these samples allows us to use DNA sequencing to determine at least a big portion of what is in the water column and what's available for food. And we can do that in different places and compare and see whether the food availability differs between sites.
We could even go at 0.1, unless you want to skip ahead.
I could cut back and forth.
I think we're running out of room to skip.
Okay.
Nice and slow is fine with me.
And this is a sampling that Okeanos hasn't really done before. This is new for D2, and this is just a demonstration to see whether this is useful for scientists and whether it impacts the operations of the whole
bridge. This is ROV now.
ROV mapping and everything else. See whether it fits in and whether scientists would like to see more of this.
15 meters, that's 15 meters, bearing 190, speed decimal 1 knot.
And one more mystery that might be solved by looking at the DNA in the water column is whether there are larvae present of any of the organisms that we see.
Copy, bridge, thank you.
Looks like flowers.
Yeah, this does look like a big bouquet. It's awesome.
Mums.
Yeah, mums. The conger eel that lives underneath that has quite a lovely home. Very well manicured and decorated.
He got Mound of the Month award this year, I think.
So after this move, Levi, I think time might be well spent just kind of moving off at a tether length to one side and then going off to the other side and looking over both sides of the peak.
Okay.
If we go too much further, we're going to be moving down slope again.
Copy that.
Would we ever do like a photo mosaic of something like this?
We could. Not on our own. We would, need someone to request it.
Yeah, definitely.
Take a peek off the west side here.
Only about 80 to 90 miles from shore here.
Definitely not the lush Lophelia banks that we have seen in the other 3 mound dives we've had.
Yeah, I'm kind of surprised. I thought that there would be a lot more live coral on the top here.
Certainly the anemones have taken hold. I doubt they would have killed off the Lophelia, but they're taking advantage of the lack of live Lophelia.
I think we'll just, I think going back and forth on each side of the ridge, and then we can put in small moves forward, and then you kind of go side to side, and Lars could even look, he could turn his head.
It just seems like great habitat for
to each
Lophelia.
side and let you go out at a tether length or something.
Go.
I think that just...
Maybe it does have something to do with the water chemistry, so it's good we just got some of those water samples.
And that there'll be another cruise here coming to this site next month to study that in much more detail than we will.
Enhance.
Yeah, we need an enhance button. Sounds good.
Thank you, Bob. I have you there at 75 degrees.
Copy that. Hi, video.
Hello.
How are you?
I'm doing good.
Good.
How about you?
Pretty good. Just
good.
Cutting back and forth with the last.
Copy that.
40 minutes. If you see something, let me know.
I definitely will.
So we're coming into the last 10 minutes or so of our dive.
Watch lead, we have 24 minutes remaining on bottom.
Great, thank you. We have 24 minutes instead of 10. That's wonderful.
That nice. These shallower dives are longer dives.
Oh, that's right.
That's nice. And a little more bottom time. In this case, a lot more bottom time. Our deepest dives, we were only on the bottom, what, 4 hours? A little more than 4?
Want to do a current drift test? Dan?
Yeah.
See what the current is?
Sure.
I'll put my
2
head
hours
into it,
to
we'll
ascend and descend. So that took a big chunk out of the dive time.
All right, watch lead. We'll do a little current drift test, just so I can, if you're interested. See what the current magnitude is here.
That's great.
Okay. All right, here we go, Dan. See what we get. Oh, it's kind of current.
There's a squid in the water column ahead of us or something going off, right?
Yeah.
Let me know when you think you're locked in.
Can snap video.
Or just mucus. That's wishful thinking.
Yep. All right, Dan, what do you got?
0.28 knots, and it's seems to be just bumped up to 0.3. It's probably pretty close.
Okay.
About 0.3 knots of current.
You can put being a reporter on your resume now. Letting us know what the current events are.
It's good. Very nice. Just in case you didn't copy watch lead, we're looking at about 0.3 knots of current moving from southwest to northeast.
Great, thanks a lot. Roger that.
Give you a 10-meter bump.
Yeah.
I already there at 6-0.
Copy that, co-pilot.
Yeah, and I would you know, just keep moving back and forth across the top of the ridge.
Sounds good, Dan.
Bridge, this is RV nav. Yeah, we'd like another move, please. We'd like 10 meters. That's 10 meters, bearing 190, speed .1.
Oh, there's a little tiny sea star on a sponge right at the bottom above the spot lobster.
Wow, that might be the tiniest sea star we've seen the whole time.
Okay, I got it. Hold on.
Good eye, Annie. Just spotted this little sea star on a Vasella sponge like the one we just collected.
Copy, Bridge, thanks.
Just getting you a stable here video, and then we'll let you come in.
That might be the cutest
partial.
Cutest one we've seen.
Mm-hmm.
And then you can take the rest of the zoom.
Oh,
nice shot.
They could tell we were coming close.
Oh my gosh.
Wow, that's
partial.
It's adorable.
You coming further?
That's full.
There you go. It's pretty small.
That's got to be a baby.
Must be the shark.
I can't imagine that's full grown.
Yeah, it's star on his chest.
It's a sponge eater, Tina says. I don't know if she's serious. I don't know if that exists.
I think we've seen some. We saw a sea star on a hexactinellid glass sponge. Well, this is another hexactinellid, but on a different, the more lacy vase sponge.
Right. I actually mean these because they're so.
Yeah,
they have so many spicules, and it's so tiny. And I guess Chris Ma, Tina mentioned that Chris Ma said the other day that they are eating sponges.
Yeah, Lars, you could just turn your head to follow him as he moves over.
Copy that. Coming to port.
And Nolan wrote that that sea star was so small and its texture is very unusual, and that it makes it difficult for him to identify a juvenile asteroid physical characteristics. So you would need to be able to see it clearly. And the underside would also help, no doubt, in its identification.
Still have squids around with inking.
There's something waving around in the current up above the squat lobster to the right.
Oh, yeah, like a translucent mesh or...
Put an egg set.
I'll get in closer.
I hope it's not more debris.
You can come in partial.
Looks like plastic. Is it a kite once, maybe?
Maybe the sticks are rolled up portions of the plastic or something. I don't know. It's like trash to me, I think. Maybe?
Yeah, it's trash.
Yeah.
I was wondering if it could have been a kite, and maybe that's what the longer pieces are.
All right, video. Pause there.
Yep.
Most people think of plastics as floating all the time, but they do degrade to a point where they sink. So they're still in the environment, even on the sea floor.
Copy bridge.
Wow.
Should we just try another 15 at where you're pointing now?
Chris Mott here.
Yeah.
Hi, Chris. Did you see that little baby sea star?
145.
Yeah. I just saw it. I hope you're not already done.
We are past it.
We are past it, yeah.
Is there anything you can tell us?
Well, but you're still on the bottom?
Yes.
Bridge, this is already
Okay.
nav.
The best that I can come up with is that
We'd
is
like 15 meters.
a strange little sea
15
star
meters,
called
bearing
Marginaster.
145.
Huh.
Speed decimal 2.
I know. Marginaster has been problematic for a number of years because it's actually every specimen that's been collected is really tiny, and so there's been a debate among people studying them whether or not it's an adult or if it's a juvenile form of something else. And what the argument against it being the juvenile of something else are simply that we've never found what it grows up into.
Huh.
And Marginaster is actually a genus of sea star. It's a perenniid.
We'll grab it.
And so it's in the same family as that large purple sea star that you guys were seeing earlier.
Huh.
So this, in one sense, complicates that whole discussion because now it's possible that that could actually be the juvenile of that big purple perenniid that you guys were looking at, rather than the idea that that others have subscribed to that's simply the adult.
Zoom further if you'd like.
So, and in answer to your other sort of line of inquiry about sea stars feeding on sponges,
we've gotten this before.
Sponges are a plentiful food source for sea stars. We saw plenty of different taxa feeding on them in the Pacific. Goniasterids, parasterids, Henricia. Henricia especially is pretty well established as a predator on sponges. So they're not
maybe it looks similar,
as sort of eye-catching as some of the like octocoral predators, the ones that are seemingly suspended in the branches of the really big octocoral colonies, but there's definitely a couple. There was I think a Henricia on that large van-sized rosellid sponge that was in the Hawaiian Islands last 2 years ago or so.
Mm-hmm.
So there's definitely a lot of.
Is there any other shots you'd like of this? Or?
How would you call it? A peripherovorous sea star. So, but yeah, that one. Just from the outline, that would be my guess. I'd have to look at the higher res image in a little bit, but there's very few things that that could be. So I'm looking forward to seeing the better picture of it. But all things considered, I'd be pretty comfortable calling that Marginaster. I think the species in the area is called Marginaster pectinatus. So, but good catch by Annie, and great visual by the camera. So thank you.
Great. Thanks so much for telling us more about that. We were totally convinced it was a juvenile for sure, and that was it.
Well, it's always a question, isn't it? Is whether or not it's. I mean, your question is literally one that has echoed the taxonomy of this species for probably the last 50 years or so.
Yeah.
Anyway, have a good remaining time on your dive, and I'll catch up with you guys tomorrow probably.
Okay, great. Thanks for joining us, Chris.
If not already.
Have a good dinner. Bye-bye.
Bye.
Thanks, Bridge. Yeah, the move is complete.
Copy that.
That's right. Tonight is the ice cream social night.
Oh, it's Sunday. That's
yeah, it is.
Right. And while Chris was talking, we saw another of the sea stars, the goniasterid variety, that we've seen several of members, or really 2 species of those. He may come back on. We thought we'd let him go for a minute. Where is he calling from? Do we know?
Washington, D.C., I think. Well, he works at the Smithsonian, so somewhere in that vicinity.
I guess it was just that he was in Japan earlier in our cruise?
Right.
Okay.
He was at an echinoderm
conference, that's right.
Got a tail hanging out of that one coral rubble bush. Imagine that is attached to a conger eel. I would love to see them building these little caves that they live in.
Would you like me to follow you around, pilot?
Or? No. Thanks.
Copy that. Have you there at 90.
Okay.
And Nolan identified the goniasterid sea star that we saw just a few seconds ago as a Plinthaster dentatus. And that's one of the
time we have left, Dan?
2 species we've seen of the goniasterids.
I wonder what the coverage of anemones is here. What percentage? It's got to be so high.
Oh, yeah.
Good fire.
Look at that in the background.
We didn't see as many when we were deeper on this feature. But yeah, just this one spot, there must be 100 at least, or more.
And the Venus flytrap, larger anemones in the foreground.
Watch lead.
Seen a lot of them as well.
At 70.
Go ahead, Nav.
Just letting you know, we have 5 minutes remaining in the dive. Would you like us to go ahead and capture the last 2 Niskin samples?
That would be great. Thank you.
Copy that.
And if you all didn't hear that, Nav just reminded us there's 5 minutes left in the dive. We're going to trip the last 2 Niskin bottles, sample 2 more approximately 2-liter samples of water.
When you're ready, Lars.
Copy that. Resetting. And firing number 4. 4 is fired. 2006 UTC. Resetting the last one. And firing number 5. Number 5 is fired as well. 2006 UTC.
When we get a chance, could we kill the lasers?
Yeah, absolutely.
Lasers coming off.
Oh, yeah.
Thank you.
Do you think this is the first time we've ever had a Spec 10?
We got up to Spec 12 the other day.
Did we?
Yep.
Okay, darn.
We still have one more open rock box.
But no
Let's
rocks.
not give them any more ideas.
I have video. Can you look off in the distance? Look at all these things. Crazy.
Looks like it feels the
there's only
dandelions.
2,
Right.
10 and 11.
All right, keep going.
Did we have a spec 11, Annie?
That was a good question.
This might be a new record.
Yeah.
Does that mean we're the 5, 10? Do we have 11?
Yep.
So we have 2 bios and 1 bio or something?
Yes. Yep.
Okay, so our time is ticking down now. We only have a few minutes left on the bottom. We'll hope that some of you will call in to our dive planning call immediately following the ROV leaving the bottom. We're going to talk about dives 11 and 12.
This is incredible. I'm thinking if somebody just tuned in, they would think we were looking at a garden of chrysanthemums. These are actually anemones. Hundreds, thousands of them. And we see almost no Lophelia here. We saw more on the flanks when there were far fewer anemones.
It's interesting how every one of these coral mounds has some similarities but some unique characteristics. And
the ones we've been diving on.
All the mounds that we've been to, yeah.
So Lars, we just have a minute or two remaining.
Different species become the most abundant. Some species are totally absent from different mounds. Some occur everywhere.
In the 6-8.
This one has attracted a lot more fish than the ones that we observed, at least in the southern portions off of Florida and Georgia.
No, well.
Maybe it's because it's a weekend.
Right now you have a turn.
I don't know.
Which means you are turned to port relative from your starting position.
Sorry, go to starboard.
Go to starboard, 0.2 turns, which will line you up.
Gotcha. So just go to...
So this area, we saw our first Eumutida picta of the cruise. So those weren't on other Lophelia mounds. And then we saw our first conger eels, and of course our first wreckfish.
And then pilot would come
our first green eye, which was one of the fish we saw at the beginning. Yes. That was beautiful blue and green color.
55550.
And we've seen the Venus flytrap anemone, but we have not seen anywhere near this abundance of anemones anywhere else.
Yeah, Vito, come in.
And this Phasella sponge is very common, very dominant here for the sponges.
All right, after this shot, we'll call it the end of the dive.
Great parting shot.
Watch lead, just so you know, after this shot, we'll have to set up for recovery.
Roger that, thank you.
Let me know when to come out.
Yeah.
Okay, so this is our parting shot. Maybe he'll wave goodbye.
Wouldn't that be great?
Or maybe he's kind of saluting already. He's saluting. The one antenna salute.
I think he's telling us it's almost dinner time.
Mm-hmm. So thanks to everybody who joined us today in the chat room and online, on the phone line.
All right, Vito, you can one.
And we hope that you'll return with us tomorrow morning for another great day of Deep Sea TV.
We're not 100% sure that we can promise where we'll be tomorrow. We have to evaluate where the Gulf Stream is. We'll be doing some mapping tonight to better define a location. So you may be surprised when you tune in tomorrow, but wherever it is, it will be an exciting dive because all of these areas have had plenty
maybe you wanna
that we can talk about, observe.
What you need him to do and all that.
Yeah.
Great video.
You can haul in on the winch.
Each site is different.